All right, welcome back. This is going to begin a two-lesson series on cell communication. So cells, just like people, need to communicate. They need to communicate with one another in order to coordinate their actions and their development to maintain homeostasis for a multicellular organism. They also need to communicate, believe it or not, with themselves in order to change and maintain their internal environments to be able to maintain homeostasis for that single cell. All right. So I'm starting off this presentation. I have a picture of a, a vending machine and this fake bill because they are a really good metaphor for cellular communication. This vending machine requires a certain signal to dispense the chips or the food inside. And that signal is this dollar bill. So the dollar bill goes into the slot, and at that point, the vending machine has received the signal that somebody wants to order something. By putting them in the money, it's received a signal. It then takes that signal and maybe the choices you select, the buttons you press, and it that signal travels from the bill receptor and the buttons to the specific little coil that holds the snack you selected. And then as a response of that signal, of you pushing in those buttons, something happens. The individual snack falls down and is available for you to take. So a response. So this simple analogy of a vending machine and some money is a really good overview or an example of how cells communicate. There's some sort of stimulus or a signal. The signal is processed and transduced, moved from where it's received for where its response can happen. And then third, a response can occur. Something can occur because that signal was received. In this case, the snack falling out and you being able to grab it. So within multicellular organisms, cells must communicate one another to do many different activities. A signal transduction pathway is a series of steps by which a signal from one cell's surface is converted into a specific cellular response. So a signal transduction pathway is a specific part of this communication sequence of steps that takes this signal when it's received by a cell and converts it into a specific response. Now, these pathways are very similar in all organisms. So even organisms as different as unicellular yeast and multicellular mammals pretty much have the same methods of cellular communication. So these communication techniques or these pathways evolved very, very early in life's history and then has been preserved for millions of years. Now, cells communicate really in three different ways. They might do it through direct contact, so cells right next to one another might communicate via receptors on their surface directly interacting with one another. They might interact in local communication, so cells in the same tissue type near each other, but not necessarily touching can communicate. And then also signals might be sent a long distance from one part of the body, like your brain, to another part of your body, like the kidneys. And this is long distance signaling where the message has to travel a long ways and travels in between cells of completely different types to carry this message. So here's some examples of cell-to-cell communication. Um, So, in certain cells that are right next to each other, like these two animal cells or these two plant cells, signals can travel directly between those cells. So notice in these two animal cells, their plasma membranes are almost are like connected. See, it's like a dotted line. There are little holes or perforations. That's called a gap junction. In some cells that are directly touching, their cell membranes have these little pores between them so that the cytoplasm of one cell is directly connected to the cytoplasm of the adjoining cell, with these little holes being primarily made of protein channels, the same type of ones we looked at in last unit that maybe work with facilitated diffusion. And so substances or messenger molecules can directly travel from cytoplasm to cytoplasm of two different cells right through those gap junctions. So, as an example, when your heart contracts, it contracts by individual cardiomyocytes um, contracting in synchronicity. They can't just all, contract whenever they feel like it. There needs to be a rhythm. And this rhythm or this rhythmicity is established by signaling molecules moving through these gap junctions in animal cells. Plant cells have something similar to gap junctions, but not in plant cells. Not only are these channels through the plasma membrane, but also the cell wall itself, like in this picture. Those junk are gaps between the two cells that are plant cells like this are called plasmodesmata, and they function the exact same as they would in an animal cell with gap junctions. They just also occur between the cell wall also. Here are two cells that instead of gap junctions or plasmodesmata have uh, protein markers, that's these green ones, and receptors, that's these purple ones, on their membrane. And when they come into contact, because these two cells are very close, they might trigger a response. See in this cell with the purple receptors, a response is triggered because the protein markers on this green cell have interacted with its receptor. So again, here's an example of a plant cell that using plasmodesmata to send a signal or a substance to fuse from one cell to the other through these gaps in their cell wall, similar to the gap junctions that exist in animal cells. All right, now on the next scale bigger, maybe two cells are not directly next to each other, but they're in the same area of the body. They're in the same type of tissue. So, for example, one muscle cell to another muscle cell, or one liver cell hepatocyte to another hepatocyte. They're not next to each other, but they're the same type of tissue and the same general area. This is an example of local signaling over a short distance. Um, They might secrete into their environment through exocytosis. Remember exocytosis? Here's a cell secreting signaling molecules in its nearby environment, and these might be something like growth factors, which are stimulating maybe these other cells that are very small or very young to grow. And so these growth factors are only active for a very short time, so they only work over a very short different distance, and they cause changes only in the same type of cells, so other liver cells, hepatocytes, maybe like this picture. So now we can't have direct movement of substances from cell to cell through the gap junctions or through surface receptors like that because they're not touching anymore. So now we need to have molecules that are serving as signaling agents that travel through the intracellular fluid to other cells of the similar type in the same area, and that's local signaling. Finally, there's long distance signaling, and long distance signaling usually involves the message traveling a long distance, a long way within the organism's body, or particularly the signal is released by one cell and received by a cell of a completely different type. So examples of this might be the nervous system in animals, so the electrical signals traveling through neurons all throughout the body, the endocrine system in animals, so hormones are secreted. Hormones are molecules, chemicals that are secreted into the blood by one cell and travel through the blood to target cells that might be a completely different type of cell, right? Your pituitary gland, for example, releases and the pituitary gland is in the brain, releases hormones into the blood, and they might travel and have effects on things like your bones and your kidneys and um, your heart and your lungs, and so various other types of cells that are far distance away from the pituitary gland. Plants also have hormones, and some transport through their vascular tissue, and others even travel through the air and have an effect on completely different plants. So, for example, acacia trees in Africa are some of the favorite snacks for giraffes. And acacia trees, when they are getting eaten by giraffes, will release a hormone that will cause the leaves in the acacia tree to accumulate tannic acid, which makes them taste very bitter and non-palatable to the giraffes, and so the giraffes don't eat them anymore. Well, when that happens, some of the tannin molecules, some of those signaling molecules that signal the release of an accumulation of tannin in the leaves, are sent through the air and are sensed, received by other acacia trees. And those other acacia trees, even though they aren't being eaten by the giraffe, produce those tannins as a preventative step because, oh, there's a giraffe nearby eating one of my neighbors. So that's a crazy far distance away, and also an example of long distance signaling. So local and long distance signaling, because they are communication between um, cells that aren't right next to each other, both use messenger molecules. And these messenger molecules, like these dots here, we also call ligands, 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 right? And um, they are an example of when one cell releases a ligand and they act on a nearby target cell. And so this target cell receives the ligand binds to probably a surface receptor on this target cell and causes that cell to change in some way. Maybe it's a growth factor and causes it to grow, right? Nerve cells do the same thing. They release neurotransmitters, which would be the ligand in this case, and they stimulate the target cell, the next neuron, to do something, in this case, to depolarize, to continue the electrochemical signal that makes us able to feel and transmits different information all across our bodies. So in this last example, notice that in here, the signaling molecules, the target cells were all nearby. They were close by. They were a local signal. We also call local signaling paracrine signaling. So local or short distance signaling, another word for that is paracrine signaling. That's to be contrasted with endocrine signaling, which is long distance through the blood, through the air to completely different cell types. In us, hormones are one of the main classes of molecules that do long distance signaling, endocrine signaling. This is why we classified, when we were talking about in our first unit in chemistry of life, this is why we classified steroids as signaling molecules, because many steroids are hormones and work in the endocrine signaling pathway. And these hormones or endocrine signaling molecules are produced in specialized cells. And when they're produced, they travel a long distance, usually through the circulatory system to the target cells that might be in completely different areas of the body. So here is an endocrine cell secreting maybe a hormone traveling through the blood, and maybe here's a target cell in the reproductive system that's responding to those hormone signals. Now, in general, this whether be local paracrine signaling, long distance endocrine signaling, or even uh, cell-to-cell contact, what we call juxtacrine signaling, with cells right next to each other, we can model that signaling, those signaling steps, the steps to produce, receive, and do something about that message, in three stages. So there are three stages at the receiving end of a cellular conversation. So the cell might receive that message, that ligand, that hormone, that signaling molecule. The first thing that happens is a reception phase. So the signaling molecule or the ligand binds to the receptor. The signal is received. The transduction step, that signal moves into the cell and throughout the cell from where it was received to where something that can happen because of it. And along the way, it's becoming stronger, it's amplified. And th three, the response phase. Something happens in the cell as a response to receiving the message. Now, for our first lesson, we're going to focus just on the reception part, this reception part. So in the reception part or the reception phase, our signaling molecule, which we also call a ligand, and here's that word again, ligand, binds to the cell surface receptor, just like a hand in a glove. So the signaling molecule is many times a steroid or a protein. And the receptor, just like receptor proteins we talked about in last unit, is also a protein. And the receptor protein has a specific binding site that's perfectly shaped for the ligand. So these receptors and these ligands are specific. A specific receptor can only bind one or only a few ligands that have the specific shape to fit into their receptor site. And vice versa, a ligand can only bind to receptors that have a specific shape so they can fit just like a lock in a key, just a key in a lock. All right. Now, when this ligand binds to the receptor protein's intercellular domain, so this part that's sticking out into the cytoplasm, its shape will change, and that shape change will then pass the message on into the transduction phase we'll talk about in next lesson. So in the step one, the target cell detects that there is a signaling molecule, a ligand coming from the outside of the cell. The ligand is detected when it binds to a protein's surface, the cell's protein surface receptor, and the signaling molecule can only fit in a specific receptor. So it can only bind to a specific receptor. So here might be a protein on the surface, and here is a signaling molecule that's binding to that protein, and that protein, that surface receptor is activated, its shape and the intracellular part is changing, and so it's able to launch an intracellular response. Ion channels are a very important type of cell surface receptor that is used in this reception phase. For them, when a ligand binds to the surface receptor actually is a closed channel protein. And so when this ligand binds to this surface receptor, it opens up, its shape changes, so it opens up and a hole or a passageway forms that allows maybe the facilitated diffusion of ions to take place. And then when the signaling molecule degrades, the channel closes back up and doesn't prevent any more and prevents any more facilitated diffusion. This is an example of like what happens in nerve cells with neurotransmitters, and we're going to learn more about that in our assignment today. There are also intracellular receptors inside the cells, sorry, uh, protein receptors inside of the cell. So when we have signaling molecules that are like small hydrophobic molecules that are able to diffuse through the plasma membrane, the receptor doesn't need to be on the outside of the plasma membrane. It can make its way in through a simple diffusion and activate a protein receptor that is already inside of the cell. So this happens in certain situations, like I said, when our signaling molecule is a small hydrophobic nonpolar substance that can get into the cell on its own. One really common kind of type of cell surface receptor is called a G protein receptor. It's very common. In fact, about 75% of pharmaceutical agents, therapeutics, medicines work by interfering with and changing the binding or the activation of G protein type receptors. Now, you don't need to memorize this, but I thought it's a good example of a general uh, step process of protein reception. Not just in G proteins, but in all cell surface receptors. What might happen if the signaling molecule stays bound to the surface receptor and never degrades and never detaches? Well, what would happen is your signal would continue to be sent continuously, far after maybe it needed to be, because the signaling molecule is never detaching from uh, the receptor. And certain mutations can occur where that is exactly the case. In addition, certain mutations could occur where this surface receptor or the signaling molecule's shape is altered. So now they can't fit together. And even though the signaling molecule is being sent, it's never, the reception phase is never occurring. The ligand is never binding to the cell surface receptor. And so no intracellular response can take place. So start to think about what could changes to this uh, reception part cause if the signaling molecule stays bound too long, or if the shape of the receptor changes, or if the receptor isn't present on the plasma membrane at all, or if the signaling molecule's concentration decreases. Those are the type of things that on the test in class and in the AP exam you're going to want to know. So start to think a little bit more critically about, okay, I understand this part about signaling molecules and receptors and then binding. So how could that change if, how could that sending or receiving that signal change if different things interfere with that binding, or if it happens and it occurs for too long? All right. We are going to stop at this point, and next class we'll talk about the transduction and the response phases later on.